So thank you very much. It's a great pleasure to be here in Milan. Delighted to be able to talk to you this afternoon. So I'm going to talk about matchmaking and the challenge of rare and novel variants. And Cambridge is a famous university city and has a great museum, the Fitzwilliam Museum, which has a particularly fine collection of porcelain. And a few years ago, a visitor unfortunately smashed several incredibly valuable late 17th century Chinese porcelain vases into smithereens, and 3 of these vases were smashed. So there was pieces of porcelain all over the floor with no one really knowing which vase they belonged to. And I think all of us working in rare disease are slightly in that position where we each have a fragment and we don't know quite which vase it belongs to. And so I'd like to talk about matchmaking and the way that we can, by sharing, actually try and resolve some of these issues. So one opportunity is through the MatchMaker Exchange, which is a federated network of platforms that promote sharing. But in order to access MatchMaker Exchange, you've got to go in through a portal, through a database. And so I'm going to illustrate that with talking about the DECIPHER database. I'm going to talk and show you how you can matchmake within DECIPHER, and then beyond DECIPHER through the MatchMaker Exchange, and then try and further the lecture we've already had, illustrating just how fantastically important these rare diseases and these rare patients are in contributing towards diagnosis and discovery. So, the challenge of rare and novel variants. So, all of us have 6 billion letters in our genome, and the scale of the interpretation problem is massive, with each of us having between 4 and 5 million variants in each of our genomes. So people have calculated that there are about 9 billion SNVs are possible. And identifying matches and conflicts leads to discovery and safer diagnosis. But how do we make that happen at a practical level? So this was a paper produced very recently, last year, showing the scale of the challenge of rare missense variants. So these are variants. Many have been discovered, but I think the concern is there are many more yet to be discovered. And most are variants of uncertain significance that occupy this huge void between the likely benign and benign and the likely pathogenic and the pathogenic, this huge space here. And in this study, Streeter et al. Plotted the number of registered tests against the total number of missense variants in ClinVar. And people might have expected, oh, well, the genes we know about for a long time, they'll be the ones we've sorted out, and there won't be so many missense variants. But in fact, it turned out that the ones at the top of the tree with the largest number of variants, albeit in quite large genes, are actually BRCA1 and BRCA2, which are some of the most tested genes. And not only did they have the greatest number of variants of uncertain significance, but also if you look at the darkness, there's a gradient increasing darkness as you go up this score. You had a greater chance of having conflicting reports the more variants there were. So these are real challenges for us working in clinical diagnostics. And so I think the need for matchmaking is not a transient phase. I think we're going to be in the business of matchmaking for a very long time to come. So it's really important that we invest in good tools and good strategies at this juncture that will serve us well as we go into the future. So one initiative that's been set up by the Global Alliance for Genomics and Health, GA4GH, was how do we find all these one-offs and data that's in silos and federate those databases into a searchable network. And MatchMaker Exchange is one of the driver projects for GA4GH. And this is the federation that's been set up. DECIPHER was one of the lead projects and has been contributing very actively to the software development that underlies MatchMaker Exchange, but it now links a lot of US, Canadian and Australian database, and DECIPHER is the European contributor to this. You can't access MatchMaker Exchange except through a portal, so you need to go in to the system through an existing portal. And these are all the different portals with their various pluses and minuses. And for the sake of argument, I'm going to take you to DECIPHER because it's the European representative here, and show you a little bit about what DECIPHER can do. So DECIPHER shares open, or it has data in three phases. It has private data that's personal to the individual project. It has consortium level data where you have a network of projects that have agreed to share data, but it's only shared within that network, like within the NHS or within the DDD study or within New Zealand. Or you can have open data where there's been consent to share from the patient, and then it's shared openly. And through that mechanism, we share all the diagnoses we make in the DDD study, and also all the interesting variants we find in genes which don't yet have a known function or role in human disease, so losses of function variants or homozygous LOFs or de novo variants. So DECIPHER was set up in 2004. It's been going now for 14 years. It's always been a platform that has sought to match the depth of the phenotyping with the quality of the genotyping. And we share just sufficient information to enable the variant to be interpreted. So we're not sharing VCFs or exomes, we're sharing the candidate variants associated with the phenotype to enable the phenotype to be linked to the variant, and with the aim or the long-term goal of mapping the clinically relevant parts of the genome. So it's a platform where you can deposit your plausible pathogenic variant and your phenotype. You can use the platform, and I'll show you how to do that, to annotate and visualize variants. You can use the platform to share data and make it openly accessible, and then hopefully to find matches, and then to collaborate and drive forward our understanding. So DECIPHER's been hugely successful in actually has provided data that's been referenced now in over 1,500 publications. So this is the collaboration network, and I've focused here on Italy. And you can see that Italy has 22 projects with over 3,000 open access patients. But that's part of a network of nearly 250 projects across the globe, with a total of 75,000 patient records and over 200,000 phenotype observations. So there's really a huge amount of data within this project now. So where do users of DECIPHER come from? Well, they come increasingly from literally all over the world. And even over the last few years, we've watched South America and Africa sort of light up with increasing usage. And in fact, it's just the Saharan area in Africa now that's seemingly not accessing this data. And you can see that Italy's quite a keen user of DECIPHER with 8,000 visits so far this year. So much of the functionality in DECIPHER is free and open for you to use without a login. You just simply go to the search box and put in a gene name and find the open access patients that are known about in DECIPHER, and much of the functionality that I'm going to demonstrate to you is actually then directly usable for you, including contacting the submitting clinician. So please bear with me, I'll talk you through how to do that. But if you want to actually use the MatchMaker Exchange or to deposit data in DECIPHER, then you need to join DECIPHER in order to facilitate that. So searching is here using, as I say, you can use a gene name, you can also use a genomic position or HGVS code, or if you're logged in, you can use phenotype terms. And joining is simple. Just click on the button here, and it will open up some information about joining the project. And then if you have an existing project in your centre and you want to join it, you can join as an established project, but you may want to innovate and join as a new project. And we're a network of academic clinical genetics services, and so we just ask for evidence about publications and university affiliations and the hospital, and then nominated contact details. And that's all it takes to set up a project. So once you're in DECIPHER, this is the sort of utility that you get. So here is a variant in CDK13. If you put the variant in using HGVS or genomic coordinates, the rest of this is generated, but also you can add inheritance and a statement as to the likely pathogenicity. And I'll come back and show this matching patients schema in a short while. But you can see it can drill down to really quite a lot of detail on these patients, including a lot of depth of phenotype about them. And there's also a lot of information about phenotype with Chris Nellicott, who's at the back of the room, has kindly worked with us to help us develop these composite faces within DECIPHER that are built up from individual images that have been submitted. We don't share those images, but we combine them to create these de-identified faces that actually provide a very clinically useful representation of the characteristic face of a syndrome. And we also can aggregate the phenotypes and look at how common they are across the database and come out with what are the most discriminating phenotypes in a given disorder. And sometimes they're not the ones you would first think of, but sometimes they're just a rare phenotype that actually is very common, often overlooked in a given disorder. But when you look at the p-values, you can rapidly generate very high p-values for some of these. So CDK13, ASD, is really present in a third of the patient, but it is a very strong marker that if you had a patient with an ASD with other characteristics of CDK13, the ASD would be a strong indicator that you were in the right gene. We've been able to combine anthropomorphic measurements where they've been added into the database to auto-generate these plots of height and weight and developmental milestones. So we can see that children with CDK13 tend to sit between the ages of about 12 and 20 months and start walking between about 2 and 4.5 years. And these plots auto-generate as soon as we've got 5 patients with an annotated pathogenic or likely pathogenic variant in a gene. So DECIPHER is automated to work across all the genes in the genome, and so it will automatically generate this data. One of the things that people have been finding most useful are the protein views, where we've worked with the team at the European Bioinformatics Institute to try to join the DNA and protein worlds, which historically have really been quite separate communities. So here we've got the gene along the top and its missense constraint scores along here. And so we've got a very high degree of constraint here with no sort of pathogenic variants identified, weaker constraint here and here, and then an area of quite strong constraint here where all the pathogenic variants seem to be piled up in the kinase domain. We've got the nomad missense variation depicted along here, and you can see that this area of constraint is really there's an underfilling of missense there. We've got the homozygous missense variants along here that are present in nomad, because that's always quite a good clue that they're not causing an early onset severe phenotype, and also the nomad LOF variants. And if this were an X gene, you would also find any hemizygous variants. So here are the DECIPHER variants. Here is our patient with this gold line. And then if we toggle down, these are the ClinVar variants. And then we can get down to where there's 3D protein structure, you get a green bar. And if we click on that green bar, you can generate a view of the protein, and you can see where the missense variants are, all on this inner face of the protein here, which corresponds to these variants here. And you can find your own, usually a patient's variant will show up as a black ball, so you can see exactly where your patient's variant is on the 3D structure. And for some genes, that's really helpful and important information. We also have tools to support diagnostic assessment, and one of the most useful is this tolerated population variation calculator, which tells you if you found this variant in nomad, is the frequency in nomad too high for it to be plausible as a disease-causing allele? And this works for both recessive and dominant models of disease. Here you can see that there's 224 of these alleles found, but observed in the population are 129. So that remains credible as a disease-causing allele. But if those were flipped round and you'd found 129, but observed in nomad were 224, then that would tell you you'd crossed the threshold, and actually this was too common to be a plausible cause of disease. We've also got variant pathogenicity, the ACMG guidelines, a calculator that will show you, once you enter the criteria, it will automatically tell you which class of variant you're in. But we've been able to implement the Bayesian classification framework that was published in 2018, which shows you where you are along this continuum. And here you can see that we're at 0.675. And that classifies the variant as uncertain. And where there's a conflict between these two systems, it will allow you to choose which one you wish to go with. It also allows you to play around and work out, well, if we tested the parents, how much would that change things? Because you can see where you move on this scale if you do that. And we've got a patient diagnostic assessment tool, which allows you to do a clinical fit. How well does this fit the patient that I've got in front of me in terms of their age of onset of symptoms, a clinical fit for the gene, the severity and progression of features and any relevant family history. And also, what, from my clinical knowledge, do I know how many other genes in the genome cause this particular sort of picture? And putting that together, you can then, there's a notes tab if you're in an MDT, and then you reach a conclusion where you choose the gene name, you choose the disease, which of the OMIM gene disease pairs you want to make an assertion about, and you state whether you've got a confirmed or likely or uncertain diagnosis, or actually it's a patient with a non-penetrant but pathogenic variant, or that you think there's no association between the gene, no plausible association between the gene and the variant and the phenotype at all. So that enables you to record the output of an MDT meeting, or really just to provide you a structured way of assessing whether you've reached a robust genetic diagnosis for a patient. So here we are within DECIPHER with a patient who we've annotated with these HPO terms using the DECIPHER tool, and in whom we've uploaded a photograph, we've uploaded some citations, and we now want to see if we can find any matches. So we've put in our variant, and I now want to use the tool to see whether we have any identical or similar variants in DECIPHER. So here's our patient. It will show us where we are on the chromosome. We're on chromosome 6, we're in the ARID1B gene, we've got a C to T change in a very conserved amino acid, and we can see that there's no identical match in DECIPHER, there's no identical match in ClinVar, there's no identical match in NOMAD, and we've got good coverage and good depth in both exome and genome data here. So no obvious matches, so we now need to look for similar patients. So if we go to the Matching Patient tab, it will bring up 59 patients with sequence variants in this gene, and it will allow us to filter across all of these different characteristics. So when we first started DECIPHER, you felt hugely pleased if you found even one other patient with a variant in the same gene. I mean, that was a huge success. Most of the time, everything was very sparsely populated. There was hardly any matches at all. But over the years, as more and more data has filled up, now the issue is, how do you tune your match to end up learning about the patients who are most similar to yours? So this set of filters enables you to do that. So first of all, we'll look at the 59, and we'll see that we've got, these are the phenotypes that are most commonly present in matching patients. These are the patients where my patient's phenotype matches that present in matching patients. And these are the features in my patient which don't match those seen in other patients. And this can be really helpful in checking whether you're in the right gene. You know, have I got a good match here? Do these phenotypes here matter? Do they indicate that they're just additional details in my patient, or do they indicate my patient got a blended phenotype and 2 diagnoses, or actually is there very little in the middle and a huge long list here, and I'm clearly in the wrong gene, and this variant isn't the explanation for my patient's features. So if you want to tune your match with filters, it's very simple. You simply click on here, and actually if you've got lots of variants, clicking on the de novo is a very good way to start, because that really gives you a very pure signal about what this gene does. And all of these lists will then automatically repopulate based on that filter. And if we want to do it again, we can add likely loss of function, because that matches our patient's variant, and then we generate this list, and we'll see that we've now got a much more precise match for our patient. We're now focusing on patients with both likely LOF and de novo, and we might decide, well, actually that's a sufficiently tight match. I now want to contact that patient's clinician. So you just click on the button, contact this patient's clinician via DECIPHER. So you click on the initiate contact button and it opens you up an email that comes via DECIPHER, and we forward on to the submitting clinician, and you just type in your message, and the message will then be sent through the system. And this is the basis for all of those contacts I was showing you on that global map. You could also decide that, well, actually, I really would like to find an identical match for my patient. So I'm going to be patient, and I'm going to click on this button here and set a watch on that variant. And then if anyone in the future enters a patient with the same variant, you will get a message like this that tells you that this new variant's been deposited, gives you a link that takes you directly to the newly deposited patient record, so that you can contact the clinician if you wish to. So that gives you lots of options for finding matches and watching variants within DECIPHER itself. But it's also, as I said at the beginning, an avenue to reach into the broader MatchMaker Exchange. So here we would go to the MatchMaker tab and click on that. And that will tell us that we're connected currently to these 4 databases, and we'll decide that we want to query those databases. And it will tell us no matches in the Broad sequer, 37 in Gene Matcher, which matches across the entire gene. It's not a tuned match, it's only across the entire gene. So you usually get a very large number of matches, Gene Matcher, none in MyGene2 and 7 in Phenome Central. So we might want to explore those. And then it will generate us a result. So you get a high score just because you've matched on the gene with Gene Matcher, but then you do get the opportunity to be able to email, but there's no phenotypes, and you don't really quite know what information is available there. And with Phenome Central, similarly, we've got the gene name, and like DECIPHER, there's information about phenotype. And I think going forward, having depth of phenotype is going to become really important, because we're reaching a stage where historically people have made a clinical diagnosis and then tested a gene. So the link between the variant and the gene and the phenotype is quite strong. But as people use whole exome, whole genome sequencing, increasingly diagnoses are being made based on the variant was found in a gene that causes this diagnosis. And actually whether the patient has that diagnosis is not clear, because you're not clear, was that diagnosis made by the lab because the variant was found in that gene that supposedly causes that diagnosis. And unless you have the phenotype, you can't assess whether you think that diagnosis is plausible. Whereas if you've got a whole list of patients with short stature and pulmonary valve stenosis, and you could say, well, yeah, that's believable. They've got Noonan syndrome. I sort of buy. But if you haven't got that information, you don't really know how much to trust that supposed diagnosis. So I think the phenotype is going to be crucial going forward, because otherwise we're in a very circular situation. So MatchMaker gives us access to data from 100,000 patients, from 3,000 contributors, and there are now 7 papers generated across MatchMaker Exchange. So really, to sum up, I think matchmaking fuels rare disease research and hugely benefits patients. And these are the collaboration requests amongst DECIPHER users over the last few years. We don't track the content of the requests, we just track whether they're happening to assess the efficacy of this system in promoting collaboration and matchmaking. So it enables collaboration, increasing the understanding of disease, and increasingly, I think this will enormously improve opportunities for therapy and research for our patients, because it's a way of making them visible and discoverable. So when I see patients in clinic and we reach a diagnosis, I discuss with them, would we like to use this knowledge now to benefit and help other people reach a diagnosis more quickly, and potentially for you to be findable if someone in Australia or the States or Spain or wherever was running a research project. Using DECIPHER, they could actually see you and contact your clinician, and then you might get invited to participate in a rare disease research trial. And I think because these trials are going to be difficult to do because the patients are so rare, that we're going to need to collect them globally, and we'll need systems such as this to be able to find them. And you can see from this map that Europe is a hub of matchmaking. So thank you very much to all of you who've contributed to DECIPHER, and very grateful for that. And it has led to over 1,500 publications. So I'd like to end by acknowledging the patients and families for permission to include their data in DECIPHER and the other databases in the MatchMaker Exchange program. The team who worked with me at the Sanger to develop and maintain DECIPHER, and the team in the MatchMaker Exchange who've enabled this federated network. Thank you.